So far, we have seen the synthesis of IMP from ribosyl 5-phosphate. Here afterwards, we will discuss the formation of individual nucleotides. One of the nucleotide was discussed in our uh, regular class. So one more is the intermediate, that is IMP, which is a common precursor for most of the nucleotides. We are taking IMP. The first step in this reaction is oxidation of IMP to XMP. So IMP is oxidized at the C2 position of the purine ring, converting the hypoxanthine base into xanthine, forming xanthosine monophosphate. So here the enzyme that is responsible for this is IMP dehydrogenase. So when we have dehydrogenase, that means NAD plus is reduced to NADH plus H plus. So this step introduces a carbonyl group, preparing the site for amination in the next step. You can see that in the six-membered ring, there are two nitrogen groups. In between that, there is a double bond O. So that is the carbonyl group that is introduced in this step by the help of the enzyme dehydrogenase. So at the C2 position, there is oxidation that is taking place. Because of that, at the C2 position, there is a double bond, which is a carbonyl group. Then the next step is amination of XMP to GMP. So xanthosine monophosphate in the presence of GMP synthetase. So here, since the enzyme is synthetase, we are utilizing ATP, and we are also utilizing one of the amino acid, that is glutamine, which is transferring the amine group. So the enzyme first activates the carbonyl oxygen at the C2 position of XMP, that is where you have the double bond O, the carbonyl group, by transferring a phosphate group from ATP, creating an acyl phosphate intermediate. So then ammonia, which is generated from glutamine, hydrolyzes in the enzyme's active site, attacks this intermediate, replacing the oxygen with an amino group to form an GMP. So you can see that where you had the double bond O, there it is substituted by NH2. Once GMP is obtained, so next is addition of phosphate group to get individual nucleotides. That is, initially we are using kinase enzyme where ATP is transferred to ADP. So here what happens, the GMP as well as the ATP, it binds to the enzyme kinase. The gamma phosphate of the ATP, that is the terminal phosphate group, it is transferred to the GMP. You can see that there are two phosphate groups in guanosine diphosphate. Then ATP is converted to ADP, and from GMP, GDP is formed. So once GDP is formed, again with the help of kinase enzyme, ATP is converted to ADP. One of the phosphate group, again it is substituted to guanosine diphosphate, that is again the terminal group. So that is how we get guanosine triphosphate. So this is about the formation of guanosine triphosphate, that is GTP.